Hey, welcome. What we are going to do in this video is an overview of the field of chemical biology to set the stage for the semester. By the end of this video, you will have a better idea about what we mean when we say chemical biology. How does it differ from other fields of chemistry and biology? How is it similar to other fields of chemistry and biology? And we'll also look at a couple of cool examples of recent studies in the field of chemical biology to get an idea about some of the directions that the rest of the semester holds in store. So let's go ahead and get started. If you wish to follow along with the PowerPoint slides that I am about to go through, you can find those by going under modules on Canvas and looking under module 1. You will find a PowerPoint that is titled Introduction to Chemical Biology or something like that. And if you download that, you'll be able to follow right along with the slides that I am about to scribble on. So let's go ahead and take a look at those as I get my screen share going here. And we'll take a look at this. So as we look at chemical biology, the first burning question is, what in the world do we mean when we say chemical biology? So we're going to tackle that right now. So when we say chemical biology, what we are referring to is the application of chemical tools to enable the study and understanding of biological systems. So when we say chemical tools that we're using for this, what we're referring to are a variety of things like chemical analyses or chemical techniques. And those could be such analyses or techniques as NMR that we'll get into in detail later in the semester, mass spectrometry that we'll also cover later in the semester, and a variety of other analytical tools or techniques that definitely fall under the umbrella of chemistry. But if we're applying those to understand a biological system, like understand how a bacterial cell copes within its environment, that fits into the category of chemical biology. So we're using these tools from chemistry to answer questions that are important to biology. We can also look at chemical biology as referring to the application of chemicals to study biological systems. So as a major example of that, when we think about small molecules, what we are often referring to here, when we think about small molecules, is synthetic compounds. So an organic chemist goes into the synthetic chemistry lab. They synthesize, for example, an array of compounds, and they use those compounds to evaluate different aspects of a cell. For example, we could be looking at small molecules that have a particular effect on a protein that is present within a cell. And by impacting that protein within the cell, ultimately the cell changes function in a measurable way. And so that would be an example of using the small molecule as a chemical probe, probing what the impact is of the protein that that small molecule is impacting on the ultimate function of the cell. So that can be used to allow us to understand the biological function of the cell, specifically getting down to the molecular level. And we're using, at the core of that, a chemical tool, a small molecule we've synthesized in the lab, or alternatively, a small molecule that we've isolated from some natural source. So the crux of chemical biology in the grand scheme of things is that it is going to be the application of chemistry to understand biology. And we will see some definite interplay there, where we will also see that understanding of biology, such as genetics, can help guide us in what chemical tools to use and guide us in using chemical tools to ultimately advance understanding of both chemistry and biology. So it's a very highly interdisciplinary area that really spans a lot of fields within chemistry and biology. Some of those include medicinal chemistry. Specifically, a lot of things in medicinal chemistry are what we could think of as chemical biology applied toward the discovery of drugs. We can think of a lot of aspects of biochemistry as feeding directly into chemical biology. Biochemistry we typically think of as just the study of the main classes of biomolecules, those being carbohydrates, nucleic acids, proteins, and lipids. But once we start applying those types of molecules toward understanding the broader function of the biological system, then we get into the realm of chemical biology. We can also fuse chemical biology with genetics, because ultimately it is the genes of an organism that play a large function in driving the overall way that the cell works. And so by understanding the genetics of a cell and merging that with chemical studies of the chemistry of that cell, we can advance the field of chemical biology and understand what is going on at the molecular level. So ultimately, we're using chemistry to advance the study of biology, and be aware there's a large gray area between chemical biology and other disciplines where there is overlap between the fields. So it's not completely possible to fully separate chemical biology out in its own field. It definitely overlaps with other areas of chemistry and areas of biology as well. When we think about chemical biology, and we're thinking about it in terms of research, there are a lot of journals that publish chemical biology research. Some of those journals are specific to the field of chemical biology, and a few examples of those journals are here. You will also find a variety of broader journals, such as the Proceedings of the National Academy of Science, Science Magazine, and others that cover the field of chemical biology, particularly when someone has a discovery that's really unique, really novel, and really hot. So when you are thinking about your video project for later this semester, one place that you might start looking as you're getting into the groove here of chemical biology is start looking through some of these journal articles that are listed here. And most of these are available through the UNF library. If you log into the library webpage, you will be able to find these journals and access articles from them. If you thumb through those articles or thumb through the table of contents, you may come across some articles that are of interest to you, and those may be good starting points for thinking about what topic you would like to pursue for your video project this semester. So it's a good idea to get started on this sooner rather than later, so that the beginning of November doesn't roll in and you're going, what do I do for my project? I want you picking a project that is something that is of interest to you, rather than picking something random on the very last day before the project outline is due at the beginning of November. So start thinking about that now. Think about what types of topics would be of interest to you. Keep your eyes and ears open. And it's a good idea to be thumbing through some of these journals to think about what topic you might like to cover. So on that topic, we're going to now look at a couple of different examples or case studies within the field of chemical biology to give you just a taste or an appetizer of what is to come later in the semester. So one example of the field of chemical biology and using chemical tools to solve biological problems is when we think about the human microbiome. So looking at the human microbiome here, that I'm circling with my laser pointer, what we're referring to there is the conglomerate of the different organisms that are present within the human. And generally, when looking at the microbiome, that collection of organisms living in a particular ecosystem, we'll be looking at one organ from the human. For example, the human gut is one common place where microbiomes have recently been evaluated, because it's been found that the human gut microbiome composition plays a major role in diseases such as Crohn's disease. Patients have microbiomes that are very different from people not having Crohn's disease. And so looking at the human microbiome, one topic of interest is tying that composition of bacteria to the chemistry of the system and understanding how the chemistry of that system, the molecules that are being produced by those bacteria, impact what the function of the bacteria are, and maybe even what bacteria are present within that system. So one way that has been explored, how we've connected the chemistry of the microbiome to the biology of the microbiome, is by starting with the isolation of the so-called metagenomic DNA. Right here in this flow chart, and circled there in green, is a bunch of different DNA double helices representing schematically the metagenome, the genetic material from many different organisms, from the full ensemble of organisms that are present in the gut. So a variety of different species of bacteria, possibly fungi, and other microorganisms. What then happens in one way of going about exploring this is that to understand the function of those organisms at the genetic and chemical level, what scientists will do is they will take and create a so-called library of the metagenomic DNA. So the metagenomic DNA contains DNA from a multitude of different bacterial sources, for example, in the human gut. And what the library does is the library, much like a library, contains a bunch of different books really well organized and cataloged from a bunch of different sources. The library of DNA will take fragments of DNA from all of the different organisms that are present in the microbiome and introduce individual fragments of DNA into different E. coli cells to multiply those individual fragments of DNA so that we can better study them. So in other words, you could think of each E. coli colony as being a different book within the library. Each of those E. coli have a different set of random genes from the microbiome, from the genomic DNA that's been isolated from that mixture of organisms. And then from there, ultimately, what is the case is that genes encode for what proteins are produced by organisms. Proteins very commonly act as enzymes. Enzymes, remember, are the catalysts of chemical reactions within the cell. So the enzymes that are present in the cell ultimately determine the chemistry of the cell. And that all goes back to what genes are present in the cell. And so once the library is constructed, then what is commonly done is that the organisms that are carrying those genes are grown in cultures, meaning that the genes that have been introduced are heterologously expressed, meaning the genes were taken out of their original host bacteria that was in the gut, and those genes have been introduced into E. coli, and now those genes are producing their protein and enzyme products, and those enzymes are catalyzing reactions now within the E. coli cell. We call it heterologous expression because we're expressing those genes in a foreign host. And we'll learn more about that as we dive deeper into the field of chemical biology. Then from there, what one can do is look at what's the biological activity of the molecules that those E. coli are now producing, and what are the chemical structures of the compounds that are being produced using mass spectrometry or NMR spectroscopy or other tools to look at those chemical structures. So then at that point, once we understand the structure of the compound that's being produced, we can tie that back and look at specific molecules and what biological activities they have, meaning what biological functions are those molecules serving. And in that way, by understanding the structures of these compounds and their biological function, we can ultimately link that back to what role are those compounds playing in the microbiome. So we're marrying biology and chemistry here using chemical tools such as NMR and mass spectrometry to understand these small molecules and tying that into a pipeline that includes all sorts of aspects of biology, microbiology and genomics to be specific here in this example. So that's one way in which we can explore the chemistry of living systems. And this top scenario, part A here, is kind of a random way of going about looking at things, looking at the full ensemble of different genes that are present within the microbiome. Scientists have also gone about this in a targeted way, meaning that scientists look for a specific gene from an organism that they hypothesize would be involved in creating an organic molecule, or the protein product, the enzyme product of that gene would be involved in catalyzing a reaction that yields an organic molecule. So we call those biosynthetic genes because they encode for enzymes that catalyze chemical reactions. So if we have some idea from previously discovered genes and enzymes that a particular protein sequence will result in a particular chemical reaction, we can look at other related organisms and find protein sequences, or DNA sequences really, that are similar. And we can compare those to the known sequences. We can take those genes and express them in E. coli, much like we were doing up here. And we can do, again, activity assays to look at the biological function of the molecule that's being produced. and use tools like NMR and mass spectrometry to identify those molecules. So in this way, this targeted way where we're looking at it for a very specific type of biosynthetic gene and biosynthetic enzyme, we can take individual genes out of the microbiome and explore what chemistry they produce. And then we can look at that chemistry that's being produced and tie that back to its biological function. So we're marrying together here, again, biology and chemistry. Another example of the field of chemical biology is applications of mass spectrometry. We'll get into this in more detail later in the semester because mass spectrometry has been an incredibly useful tool in the study of biology. But for now, as an appetizer or teaser toward this, we'll look for a moment at MALDI mass spectrometry as a way of evaluating how bacteria interact with one another. When we say MALDI, what MALDI stands for as an acronym is Matrix Assisted Laser Desorption Ionization. I know that is quite a phrase there. That's why we use an acronym MALDI. So matrix refers to when you are conducting this type of mass spectrometry experiment, the surface that you are going to be conducting the experiment on is covered with a matrix. The matrix facilitates the formation of the ions that are necessary for mass spectrometry to take place. So that's why we call it matrix assisted, because there's a matrix deposited on the surface of whatever type of substrate you are going to be evaluating the chemistry of. Laser desorption, the LD part of MALDI, refers to the fact that in order to eject ions from the surface of the organism that you're evaluating, a laser will be used. And then ionization is referring to the fact that anytime you are doing mass spectrometry, ions are required. So what's going to happen here in this MALDI experiment is that you will have two bacteria colonies here and here. And in those two bacteria colonies, what we're interested in is how they interact with one another. And being interested in how they interact with one another, one way that we can study this is through this MALDI technique. And so in the MALDI technique, if we want to look at what chemicals are present and being produced by colony A, this pink colony we'll call that, versus colony B, the one over here that's gray, what we can do is coat the plate with the so-called MALDI matrix. So it'd be coating the plate with a MALDI matrix there that I'm showing in orange. And that MALDI matrix is going to facilitate the ionization process. And so what happens then is the ion beam, or the laser beam rather, comes in like so. So our laser beam is coming in, and that is going to desorb compounds from the surface of this bacteria here that's in pink. And that compound that's being desorbed comes up, it will enter the mass spectrometer, and ionization occurs, and a mass spectrum will be generated for that particular pinpointed position of the Petri dish that we're evaluating. So right there, where I've placed a star, would indicate where that particular compound resided. And so if one continues to collect mass spectra all over this plate, just scanning all the way down the plate, then what happens is that mass spectra are collected across the entire sample, and the data can be evaluated for particular compounds, particular ions of compounds to be specific, that are present in some regions and not others. So for example, if we look at a mass to charge ratio of 701, M to Z stands for mass to charge ratio. That's referring to the ion that was present related to this molecule. Then we can look at where that molecule is physically located. Based on this heat map, it was physically located toward the right-hand side of the plate, suggesting that it's produced by the bacteria that's here schematically on the right-hand side of the plate. Likewise, this molecule that's at a mass to charge ratio of 743, showing up in green over here in our heat map, also corresponds to this circle, this part of the plate, indicating that both of these compounds are produced by the organism on the right. And we could look at concentration gradients of how the compound is diffusing between the colony on the right and the other colony to even better understand who's producing that compound. And we can also then answer questions like, how is that compound that's being produced by the organism here influencing the organism that is to its left there? What are the chemically driven communication signals going on between these two bacteria? Because the two bacteria, if they're plated on a plate together, they can't talk to one another in words like we do. Bacteria A can't say to bacteria B, get away from me, or hey, come closer. Instead, they have to use chemicals to do that. And so the biological role of this compound, as it turns out, is that it is going to act as a governor of what bacteria are able to come into close contact with the bacteria that's producing that particular compound. So it's a chemical signal that these bacteria are using. And so multi-mass spec is a powerful tool that we can use in the field of chemical biology for understanding how bacteria and other microorganisms interact with one another by elucidating where compounds are located within an environment that contains multiple different bacteria, that contains multiple different features. So we will get into mass spectrometry more later in the semester. We'll get into the nuts and bolts of interpreting some of these data and how we can even further apply this. So stay tuned for that. What we are going to do in the next few videos is lay the foundation for the depth studies that we're going to be looking at further on in the semester. We are going to build that foundation by looking at some fundamentals of biochemistry and fundamentals of molecular biology, information that you will need to know and will be incorporating into this bigger field of chemical biology. So stay tuned for that as we roll onward here into the next videos.